Our next speaker is Dr. Virginia C. Cuevas. Dr. Cuevas is a professor emeritus at the Institute of Biological Sciences, University of the Philippines, Los Baños. She specializes in environmental biology, plant fungal ecology. As a scientist, Dr. Cuevas has developed agricultural technologies. These are the trichoderma activator for rapid composting of agricultural waste and the trichoderma microbial inoculant, or TMI, for which she has obtained a patent. Her TMI is being used widely in the Philippines as crop growth promoter, biocontrol of crop pathogens, and as biofertilizer. Dr. Cuevas is a presidential awardee and was presented the Rizal Pro Patria Outstanding Agricultural Scientist Award. She has numerous awards in teaching, research, and public service. Dr. Cuevas is a member of the prestigious National Academy of Science and Technology as academician. Dr. Cuevas?
Okay, sorry. I'm still getting ready with my presentation. Okay, thank you, Dr. Cadiz, for the introduction, and good morning to all participants. My paper is entitled In situ rhizostroke composting and the use of trichoderma microbial inoculant, possible adaptive strategy to climate change phenomenon in lowland rice cropping. The data that I am going to present were generated from this particular research project ongoing, where I am the project leader and funded by Bureau of Agricultural Research under Department of Agriculture. My co-researchers are Dr. Banaay, Ms. Marisa Luna, Dr. Agam Cuevas, and we are implementing a multidisciplinary type of research. Now, situationer. Our study area is in the province of Marinduque. It's an island province between Quezon and Mindoro Island, and our municipality is Mugpog. There were two mining companies that are no longer operational today. The Consolidated Mines Incorporated and Mark Copper. CMI operated in Mugpog, but Mark Copper operated in Buac and Santa Cruz, but the ponds, the dams for tailings, which was breached in 1996 and 1993 and 1996, and inundated the rice paddies along the Mugpog River and continuously affecting our rice paddies up to the present. So this is the conceptual framework of the project. On this left-hand corner will be the environmental situation, where contamination of agricultural areas led to heavy metal toxicity that resulted in decreased crop productivity, resulting to low income from poor harvest, and resulted in exacerbating poverty for the host community. So our project intervention would be addressing contamination by remediation, using compost of plant residues, and use of microbial inoculant, which promote plant growth and control pests. We believe that we will be able to address poverty, alleviating poverty through increased productivity. And our strategy, I believe, is sustainable because we are capacitating the farmers through series of training, and we are addressing the problem of the agricultural lands. Now, this is actually the last in my series of projects, whose main activity is to rehabilitate agricultural lands that have been damaged by rock mine waste. And we have already published a series of articles on the topic of the role of compost or soil organic matter in alleviating copper toxicity and the role of TMI in increasing productivity in contaminated agricultural land. Okay, so in 2017, when we started our project, the very first thing that we studied the soil. Based on local knowledge, we were able to identify the different barangays that are affected by mine tailings. And in two barangays, we found out the contamination is very severe because even at 1 meter depth, the level of copper is more than 300 milligrams per kilogram. And these are the data that we have. There were 12 or more barangays that are heavily affected. And based on literature, if copper is present in parent material, up to 100 milligram per kilogram will be considered as normal. And since the two mining companies are extracting copper minerals from the rocks, we believe that this will hold true. So what we did was to categorize, we grouped the rice paddies into three. Copper, soil copper will be 42, and that will be milligrams per kilogram, and that will be still considered as normal. Now, rice paddies with 110 to 144 milligrams per kilogram to be contaminated, and those greater than 290 milligrams per kilogram as severely contaminated. Now, focus of our study would be the effect of rice straw compost and PMI on yield of rice in copper-rich paddies coupled with water stress. We have around 100 farmer cooperators, and our farmer cooperators are resource poor. They are mostly tenants. They cultivate on the average only half hectare. And due to financial constraints, most cannot apply the required amount of chemical fertilizers. They do not even have funds to buy fuel for the motor pump for irrigation, and this will be very critical during drought periods. In a good year, meaning to say no water stress, the mean yield in the area will be only 3.3 tons per hectare, which is way below the national average of 4 tons per hectare. Now, this will be some pictures on our rice paddies. Now, in the background here will be the CMI mined out area. So we'll have seepage from these mined out areas, and Barangay Kapayang is one of the most heavily damaged. And this is where we found that at 1 meter level, the copper content is still greater than 300 milligrams per kilogram. Our second site would be the barangays that were affected by copper mine tailings. No, so for methodology, from the 100 farmer cooperators, we selected at least 15 and made observations for 4 cropping seasons from 2018 and 2019, which are El Nino years. In the treated paddies, we have rice straw from previous harvest scattered in the paddy, and then we mixed trichoderma activator with 14, and then broadcasted this mixture over the straw, and the straws were incorporated in the soil during land preparation. The allied seeds were coated with trichoderma microbial inoculant. For the control or rice paddies, farmers practice, the rice straws were discarded and the palay seeds were not coated with TMI. We monitored closely the amount of fertilizer applied for both paddies. Farmers applied the same cultural management, and we also monitored the yield of both paddies. All other farmer cooperators implement T1, which is the treated paddies. For data processing, we computed the amount of NPK from chemical fertilizers and from rice straw using information from the literature. We have also the copper concentration in the rice field, and we categorized the water stress during the 4 seasons into 6. 0, no water limitation. 5 would be most severe, meaning to say water is limiting all throughout the cropping season. The soil in Magpog from literature is clay loam, and we use this information for computation of fertilizer recommendation. For statistical analysis, we correlated the nutrient inputs, soil copper concentration, water stress level, and the impacts of these parameters on yield. So some of our results. Now, this picture was taken 1 month after transplant, and this was taken in rice paddies where there is normal soil copper. Notice that here in the control paddies, the rice crops are exhibiting yellow-green leaves, whereas here in the treated paddies, the rice crop have greener leaves. Now, statistical analysis show that the amount of NPK from chemical fertilizers applied in both the treated and controlled paddies are not significantly different. Therefore, we can conclude that the greener leaves here in the treated paddies would get nitrogen contribution from rice straw. And we can use this as proof that the decomposition of straw was complete. Now, this is very significant because farmers do not use the rice straw because rice straw decomposes slowly, and there will be competition between soil or decaying microorganisms and the rice crop during cropping, and the leaves would show yellowing. But in this case, you will see that the treated paddies did not exhibit any yellowing. Now, this observation is carried through the grain filling stage. Notice that the rice flag leaf of the treated paddies would be much greener than compared to the flag leaf of the controlled paddies. And we have quantitative data to show this observation. Here in an normal soil paddies and then you contaminated, the leaf color chart reading is higher than 3, whereas the farmer's paddies or the control will be below 3. I have already mentioned that the farmers have financial constraints in supplying the required amount of fertilizer. Now here, LCC reading of 3 to 4 means nitrogen content of leaf is sufficient, therefore chlorophyll content is enough for photosynthesis. Now, the the control paddies would be below 3. It means the crops are deficient in nitrogen. Now, for our results. Now, yield increase of treated paddies, those in blue graphs, would be highly significant compared to the control paddies. In group 1, where the copper is normal, copper level is normal, the mean yield increase of T1 versus T sub-0 is 76%. In contaminated paddies, the yield increase of T1 versus T2 is 67%, and yield increase in severely contaminated paddies would be 150%. Now, at all cropping seasons with various stress level, you'll notice that treated paddies would have significantly higher yield. But I would like to point out this dry season 2019, when water stress is very severe at 5. Notice that the treated or the control paddy has 0 yield, whereas here in the treated paddy with normal copper level, the yield is about 2 tons per hectare. Here in contaminated, again, the control has 0 harvest. Here, the treated would be about 1 ton. And here in severely degraded soil, you have 0.5 ton per hectare in treated and 0 for the controlled paddies. Now, the other stresses, water stress and copper concentration, would be negatively correlated with yield. The effect of water stress is very significant. For every unit increase in water stress, the yield decrease is 0.064 kilograms per square meter. But this yield decrease only explains 42% of the variation in yield. It may be due to other complicating factors such as copper concentration in the soil and nutrient inputs. Now, a statistical analysis also shows that potassium is highly correlated with yield, nitrogen, and phosphorus, and that potassium contributed most to yield. Now, I have already mentioned that farmers apply very minimal of the potassium fertilizer, and so the potassium that increased yield must have come from the incorporated rice straw. Now, we have the bar graph of the nutrient inputs and pK from rice straw. Notice these red bar graphs. They stand for potassium input. And if you will recall the previous bar graph on yield, you'll notice that yield increases would be highly correlated with the increases in potassium. So our findings show that the rice straw contributed significantly to soil nutrients such as potassium and nitrogen that significantly increased yield. In addition, rice straw compost increases soil organic matter. And increased soil organic matter improves water holding capacity. And this is very significant And we have shown this in our results during dry season or during El Nino years. Increased organic matter also promotes higher cation exchange capacity that contributes to better nutrient absorption. Organic matter also increases soil pH, which makes the heavy metal like copper less available. Organic matter also chelates heavy metals, rendering them less toxic. Thus, farmers find scattering of rice straw after harvest less laborious. Now, the rapid decomposition of rice straw was aided by trichoderma activator, which was mixed with NPK, and they serve as basal fertilizer. MI contributed to rice cropping by growth promotion, protection against crop pathogen, and efficient nutrient absorption. Therefore, our results show that the technique of incorporating rice straw during land preparation with use of activator and NPK fertilizer plus the seed coating of palay with TMI can help mitigate the negative impacts of erratic rainfall pattern associated with climate change. Thank you very much for giving me the opportunity to present our results.
Thank you very much, Dr. Cuevas. We now